Okay, hi everybody. My name is Neerja. I'm a fifth year PhD candidate in the Biochemistry and Biophysics program at Brandeis University. And today I'll just be telling you a little bit about my journey through science, from where I started as a high school student, all the way to where I am today, almost at the end of my PhD. Hence the title, Almost Dr. Neerja. Before I get started, I really just wanted to say a massive, massive thank you today to both Marissa and Anahita for organizing this. I'm really looking forward to talking to everybody today. So, a little bit about me. I grew up in Ann Arbor, Michigan, which is a suburb of Detroit. It's in Southeast Michigan. Those of you who are actually in the process of applying to college may have heard of Ann Arbor before, as it's the home of the University of Michigan. And so this is a picture of my family. I have my brother on the left, and then our parents. My brother is a senior in college, and my parents are actually both academics. So my mom is a physician and my dad is a professor of applied math and engineering. They were at the University of Michigan, but they recently moved, but that's not really important. Really what I wanted to talk about was that I'm sure some of you might be imagining that because both of our parents were academics and were scientists, that they would have pushed us towards studying science when we started thinking about college. And actually, instead of insisting that my brother and I go into science or just any STEM field when we applied to college, they just naturally encouraged our curiosity and encouraged us to ask questions wherever that might lead. So there was really never any pressure on us growing up, but for me, just being around science made me naturally gravitate towards it. And so actually, when I was in seventh or eighth grade, my mom got this magazine from her medical school, and in it, at the very, very back of the magazine, because I would read these magazines cover to cover, even though I didn't understand a lot of it, it was still really interesting to me. There was this article talking about this really cool game called Eterna. And so a little bit about Eterna. It's a citizen science project, which means that anybody in the world, regardless of their background, or their scientific knowledge or lack of scientific knowledge, can actively participate in and contribute towards real life academic scientific research. And so in Eterna, the Eterna participants would actually solve little puzzles on our computers that would actively contribute towards understanding the structure of RNA. And so I took this article to my parents and I was like, hey, look, I can actually do real science just by playing computer games. And of course, because I wasn't even in high school, and here I was trying to convince my parents to let me play a video game with people from all over the world who I'd never met before, it took them a little bit of convincing. But of course, since I'm actually here now telling you this story, I was able to convince them eventually to let me try out Eterna, and that actually really opened up my world to some very exciting possibilities that I'm going to be telling you a little bit about as we get further along in my talk. So after that, I started high school. And when I was a freshman in high school, I found out about this club at my school called GUIDUS, which is short for Genes in Diseases and Symptoms. And outside of Eterna, this was really my first introduction as a freshman in high school to basic biology and computational biology techniques. And after a year in GUIDUS, I learned that it was an offshoot of an organization, a local organization called MIRCOR, which was a nonprofit that worked with local academic scientists and let me get even more experience in various computational biology research techniques. Specifically, we were doing research in microRNA and learning about microRNA expression and how microRNA expression patterns were linked to various diseases. And really, I just want to drive home that I was just a high school student when I was doing all this. And to get started to learn about all of this, all I really needed was an internet connection. So there's a whole world of science out there that's just waiting to be discovered. And like I was saying earlier, I knew from a pretty early age that I wanted to be a scientist, that I wanted to be some kind of scientist. And so these early opportunities that I sought out actually really helped me chase after that dream, even as a high schooler. And after about 2 or 3 years of playing Eterna, I got pretty bored of just solving these puzzles. I mean, there was really only so much that I could do after a certain point. And I actually, reached out to the developers of Eterna and I asked them, hey, what else can I do? How else can I'm really excited about this science. How else can I contribute to this really exciting science? And one of the other developers of Eterna, actually introduced me to a high school student that had already been working with him. And together, from Eterna was based in California and I was in Michigan, this one high schooler in California and me in Michigan applied to a national science competition that unfortunately no longer exists, but it was called the Siemens Competition in Math, Science, and Technology. And we wrote a fully original research paper as high school students, expanding on some of our results from past Eterna experiments. And when we submitted our project, we actually made it all the way to the regional finals in 2014. And so we got to present our work to a panel of academic scientists. And that was actually, and though we didn't win, it was still a really validating experience for me because it was actually the first chance I'd had to do real research. And on my first try, it got me all the way to the regional finals of this really big, really cool national competition. And so that really lit a fire in me. And I said, I want to seek out more opportunities like this. And so then the summer before my senior year of high school, I applied to a pre-college program at Stanford where I got to do even more computational biology research. And then when it came time to apply to colleges, I was 100% sure. I was like, I know I want to study biology. I know I want to do biology research. Let's see where I end up. And so I moved from Ann Arbor to Pittsburgh, Pennsylvania, where I went to the University of Pittsburgh. From the day that I started at Pitt, I knew that I wanted to do undergraduate research. I knew that I wanted to learn more about computational biology. And so within the first week of my undergraduate studies, I made a list of professors whose work I was interested in, and I just started emailing them. And of course, I told them about my background and what I was interested in researching, and I got really, really lucky. And the first professor that I reached out to actually emailed me back almost immediately, and he said, "Yes, I would love to have you in my lab. Let's set up a meeting." And this was as a freshman, my first week in college. And so I spent 4 years in this lab learning about computational drug discovery and identifying drug candidates and learning some more computational biology techniques. And because I was in this lab for 4 years, I actually got my name on a paper as a senior in college. And it wasn't like I was one of the last authors on this paper or just tacked onto the paper because I was in the lab. I was actually the second author on this paper, which ultimately meant that I had made a pretty major contribution to the actual body of the paper. And so in the words of Shia LaBeouf, what I'm trying to say here is just do it. Just go for it. If you're ever scared to take that leap, just do it. I know it's scary, but I promise you it's okay to do it scared. What really matters at the end of the day is that you're doing it. And if you know what you want to do, if you know you want to do research, there's no harm in reaching out. And it can actually really be a huge benefit to you if you reach out and start doing research early. And okay, so I did research for these 4 years. Great. Not just great. I learned a lot of science and a lot about science. But aside from the science, there was really one other important thing that I learned. And that was how important it is not only to have a good mentor, but be a good mentor. I've been very, very lucky in both my undergraduate and graduate studies to have mentors who have actively supported me and encouraged me to be not only the best scientist possible, but the best lab citizen and just person possible. And now at the end of my PhD, as I have started to teach and mentor people of my own, I feel like I have a duty to, and I want to pass down that culture of mentorship that has been fostered in me, and I want to pass that mentorship and support forward. And so I often ask myself, when I was a freshman in college, when I was just starting my research journey, what kind of mentorship and support would 18-year-old Neerja have needed to see to feel the most seen and supported? And what can I do to be that sort of mentor? And really, though like I've been saying, I've had really amazing and supportive mentors. The one thing that I really struggled with in undergrad was not seeing representation. I have been very lucky to have very lovely and supportive parents. And I have been out as non-binary actually since my first year of college. But it was kind of scary at first, right? Not because I felt unwelcome, thankfully, but just because it didn't really feel like there were other people in my college and in the spaces that I was inhabiting in these labs that were like me. And of course, that changed as I progressed through my undergraduate studies. I met professors who were out and that really were important representation for me. But ultimately, I had to go and seek a lot of those people out and make those spaces for myself. Um, and as part of seeking out those spaces, I was a peer mentor like throughout my undergraduate studies. I was very vocal about my identity and about making spaces for people like me. This is actually just a photo of me and one of my undergraduate mentees when I was one of my peer mentees. And then in my junior year of college, I actually co-founded a student organization with one of my friends, called Aquarius. This is me and one of the other Aquarius board members. And I won't go through the entire acronym because, as you can probably imagine, it is quite long. But Aquarius is and was a student organization for queer and transgender Asian American and Pacific Islander students at the University of Pittsburgh. And that was the first time throughout all of undergrad that I felt like I had a space because I had made a space that was mine, where I truly felt seen and understood and where I belonged. And I know I'm saying all of this, and some of you might be thinking, what is academia like a scary place or something? Or is it an unwelcoming place? I don't want you to think that, right? I personally actually feel as though, like, since I've started college, and since I've progressed in my academic journey, academia has actively been making strides and actively trying to be a safe and welcoming place for queer and trans people. And that might not really be apparent right away, but I think one thing we need to remember is that at its core, science is a very collaborative process. And so being able to work together regardless of who we are as people and being able to come together as scientists really is important for doing good science. And of course, as part of doing that, you will be seen and heard, and that, of course, is very important. And so then I graduated college in 2021, right in the middle of the pandemic. And when I was thinking about what I wanted to do next, I knew that I wanted to keep doing science and I wanted to keep doing life sciences and biology research. And over the course of my undergraduate studies, I became really interested in how computational biology and actual biology that we do at a bench talk to each other and inform each other. Um, I also knew that I wanted to continue making spaces like for people like me in academia to feel safe and welcome. Now, of course, you really don't need a PhD to be a scientist, right? When I was 12 or 13, when I was barely a high schooler and playing all these Eterna puzzles, I think I would have called myself a scientist if you'd asked me. But I really wanted to pursue science at the highest level, and I wanted to have my own research project and be making really cool and really important discoveries in science. Um, and so here I am now doing my PhD. I'm in my fifth year of my PhD. This is a photo of my lab. Um, and our lab studies evolution. Not just any kind of evolution, but I specifically am interested in how evolution happens all the way at the molecular level. So let's talk a little bit about what that means. So if we were to shrink ourselves down and go into our cells, we have these proteins that are called enzymes. And the really cool thing about enzymes is that their job is to speed up and do chemical reactions. So they'll bind their substrate in a place in the enzyme called the active site, which is where that chemical reaction happens. And then once the chemistry is done, they convert their substrate to some kind of product and then release that product. Enzymes do a lot of different things in our body. They're responsible for generating energy. They're responsible for cell division. Really, they perform the chemistry of life. And so our lab is really interested in asking questions about how these enzymes evolve, and specifically, how do they evolve to do new chemistry? Of course, we could really point at any enzyme in our cells and say, hey, how did that evolve? How did that evolve? And so actually in our lab, we're interested in two closely related enzymes called lactate and malate dehydrogenases, which I will be abbreviating for the rest of my talk as LDHs and MDHs. And it's actually really cool. So if you were to look at any organism across all the domains of life, all the way from the smallest bacterium to something like a blue whale, every single one of them, including you and me, would have lactate dehydrogenases. So LDHs are what are called essential enzymes, which means that without them, we would not function. We would die. So LDHs are responsible for very early steps of cell respiration, and they're pretty closely related to another family of enzymes called malate dehydrogenases, or MDHs. And these enzymes are responsible for later steps in cellular respiration. And if you just look at the molecules that they act on, they're pretty similar, right? Um, and so actually, if we make what's called a phylogenetic tree, which shows how organisms or proteins or genes are related across evolution, we see something very interesting. So everything in red here are LDHs, and everything in blue over here are MDHs. But if we look at this tree closely, there's some LDHs mixed in with the MDHs. And so you might be asking, hey, Neerja, what are they doing there? And so if we zoom in this little group of LDHs over here, these are all LDHs from a phylum called Apicomplexa. And you're probably thinking, Neerja, what are Apicomplexa? So if you ever had to take medication because you go to a tropical country and there's a risk of you getting malaria, malaria is actually caused by a single cell organism called Plasmodium falciparum, which parasitizes mosquitoes. And Plasmodium is a member of Apicomplexa. So all members of Apicomplexa are small parasites that are responsible for a lot of illnesses that we commonly get. If we look at this tree, we can actually see that all of these LDHs over here originally came from MDHs. And so because of that, something kind of weird and unique has happened in their evolution. So what we see is that because they've evolved from these MDHs, they have a slightly different amino acid sequence. And really the most important change is that they have this amino acid called lysine, where this glutamine should be. Um, and that's a pretty big change. So, one, how did that happen? Um, Two, what does it mean for the protein? Like, are these enzymes still even lactate dehydrogenases? And so one of the most important things that we do in biochemistry is we study how proteins look at the molecular level, and this is called solving their structure. And actually, one way to think about it is that it's kind of like a puzzle, a little bit like the Eterna puzzles that I was telling you about earlier. And so when we solved the structure of our Plasmodium LDH and we compared it to an LDH from pigs, we found something really weird. In the space where that glutamine is in the pig LDH, there's actually this tryptophan. And again, these are pretty different amino acids. So you might be asking, how did this happen? Um, so if we're studying evolution, if we're studying how you and I evolved or how another animal evolved, we can rely on something like the fossil record and things like paleontology to understand what our ancestors look like and how long ago they lived and how we evolved from, like, fish or apes or something, right? Unfortunately for proteins, that's a little challenging because we don't really have something like a protein fossil record. But if any of you have seen Jurassic Park, then you remember that the scientists, I guess this is a little bit of a spoiler for Jurassic Park if you haven't seen it. Since the scientists didn't really have the full genomes of some of the dinosaurs that they were trying to resurrect and put in Jurassic Park, right? They had to fill in the gaps in their sequences with frog DNA. And that had some pretty drastic consequences. Luckily for us, we don't have to do anything drastic like that. Um, but we can do something quite similar. So it's this technique called ancestral sequence reconstruction, or ASR. And basically, we use the information from our modern day protein sequences that we know, like that we know for sure, to then make pretty good guesses about what our ancestral proteins look like. And that allows us to determine how they behaved. And here's a little bit about how ASR works. So first we go to our phylogenetic tree and we get the sequences for our ancestor, and then we order the gene and use E. coli to grow up our ancestral proteins. Then we do something called a protein purification to harvest our ancestral proteins. And then once we have them purified, we can study their activity. So we can ask questions like, what kind of chemistry did these ancestral proteins do? What did their structure look like? And then we'll do that repeatedly across our entire evolutionary tree to understand and plot out how our LDHs evolved. And so in doing ASR, we actually discovered something very interesting, which is that our ancestral LDH, which I've shown on the left over here, is actually more than just an LDH. So if we look at its activity, which I've shown here on the right, um, this ancestral enzyme is doing the LDH reaction, and it's doing these other three reactions, and it's even doing a little bit of the MDH reaction. Um, so how did we get from this enzyme that was not just an LDH, but a sort of enzyme that did all of the LDH-like reactions to an LDH, which really just does its one reaction very well? So unfortunately, sometimes answering that question with ancestral enzymes can be tricky for a variety of reasons. So as I've been working on this project, I've had to pivot a little bit and look at a modern day enzyme. And if we're looking at the activity of this enzyme, you might be thinking, hey, wait a second. The activity for this modern enzyme kind of looks familiar, right? Um, it's pretty similar to the activity for our ancestral LDH. So we can use our modern day LDH in place of our ancestral LDH to understand how we got from an enzyme that does all the reactions to an enzyme that is just the LDH. And of course, I'm not done with my PhD yet, so this is still an ongoing project. But over the last few years, we now have a pretty good idea of how this evolution happens. So before I end my talk, I just wanted to give a thank you to everybody in my lab, specifically my advisor Doug, who's been instrumental in getting me to this far in my PhD. I also wanted to give a special thank you to my undergrad Maddie, who has been helping me a lot with this project. I also wanted to thank the SciComm lab at Brandeis for helping me polish up and put this talk together. And I wanted to thank Anahita for organizing this opportunity once again, and to all of you for listening. And then I also wanted to thank my cat Cicero, who let me practice this talk on him so many times and was a really wonderful listener. And with that, I will take any questions. Thank you so much.